The topic of Lecture 9 is blood plasma coagulation cascade, blood clot, also known as thrombus formation, and fibrinolysis. In this lecture, I'm using some slides which I got from a book entitled Thrombosis and Atherosclerosis. The slide numbers of my lecture are shown on one column, and next to it, respectively, are shown the pages from the book Thrombosis and Atherosclerosis, from where I got those particular illustrations or slides. I made many more slides from another book with the title Atlas of Hemostasis. In the column on the left, you see the slide number of my presentation, and in the column on the right, you see the respective pages from the book Atlas of Hemostasis, from where those particular figures came, and I used them to make the slides. Up to this point, our review of the blood coagulation process focused on the contribution of platelets. The present lecture will address the contribution of another very important participant in this process, specifically the plasma coagulation cascade. In addition, the process of fibrinolysis will be reviewed. The function of one brave activated platelet to stick to the site of damaged endothelium and thus prevent loss of valuable blood from the circulation, shown on this particular slide, may be successful in the case of small blood vessels in small animals such as a mouse or a rat. In the case of larger blood vessels, extensive damage to the vasculature, and bigger mammals, prevention of blood loss from the circulation needs the formation of the platelet plug. What that means is that many more platelets need to be activated as a result to their exposure to various chemical compounds, some of them released from the damaged tissues, some of them released from the storage granules in the cytoplasm of the platelets, and some of them activated because of de novo, from scratch formation of very potent aggregating agents such as arachidonic acid. This particular platelet plug formation is illustrated artistically on this slide. The cartoon on this slide is a reminder that what we will address in this lecture are various chemical compounds and chemical reactions. Those start with the damage to the blood vessel wall, illustrated on the upper left-hand side by the scalpel of the surgeon, which cuts through the blood vessel wall. As a result, and in the cloud underneath, one sees many chemical compounds which are released from the damaged tissues and many cells which are released also from the damaged tissues. Inside the liquid part of blood, there are many other chemical compounds which are usually in inactive form, but as a result of the damage to the blood vessel wall and the activation of the platelets, they may get activated as well. One of those cascades of chemical reactions is illustrated by the serpentine notion, which is in blue, and this is the plasma coagulation cascade. Another one is the prekallikrein-kallikrein kininogen system, which is illustrated in green. The fibrinolysis process is illustrated in red. And last but not least, the protein C activation, which brings about the activation of white blood cells, is illustrated in yellow. One thing that needs to be emphasized is that these particular processes are interconnected. One leads to the other, one stops, the other starts, and in addition, in between, one affects the other. For didactic purposes, we are going to separate each process, and we will review each process in order to understand the details of some of the important steps in this part of blood coagulation. The process of blood plasma coagulation is a cascade of chemical reactions which involve proteins which are dissolved in blood plasma. Those proteins are fibrinogen and the plasma coagulation factors. This is the reference for slide number 11. The large scale of the illustration, which appears on slide number 11, did not leave any room to include the reference in one and the same slide. The percentage composition of blood components is illustrated schematically on this slide. Focusing on the liquid part of blood plasma, it accounts for approximately 55% of whole blood volume. Inside this water environment, 7% are dissolved proteins. There are many dissolved proteins as shown on the test tube on the right. We will focus on the coagulation proteins, specifically fibrinogen and the coagulation factors, in the present lecture. In terms of weight percentage, blood plasma is 92% water, 7% proteins dissolved into that water part, and 1% other solutes such as electrolytes, nutrients, etc. Among the proteins which are pertinent to the plasma coagulation cascade are fibrinogen, which accounts for 4% of the 7% total proteins present in plasma, and other proteins, specifically the plasma coagulation factors, which account for 1% of the 7% total proteins present in plasma. We will start with a review of the coagulation factors. These are chemical compounds which in hematology are referred to as factors, and if you write the word factors, you have to capitalize the letter F. In addition, you will see that we have several coagulation factors which have been listed with Roman numerals 1 through 13. Number 1 is fibrinogen. The other ones have additional names, and for example, you notice that coagulation factor 9 is also known as Christmas factor, coagulation factor number 10 as Stewart factor, 12 as the Hageman factor. Where do these names come from? Usually in hematology, these names were given to these very important proteins when they were discovered, and the name of the patient in which they were discovered, or the name of the researcher or physician who discovered them, was given to the proteins or the coagulation factors as well. In addition, you notice that there is one coagulation factor number 3 Roman numeral which is missing. What happened is that after a comparison of the results from various laboratories, it was decided that whatever was labeled number 3 was a redundancy. It was already have been discovered and it was already given another number. Again, whenever you are mentioning the coagulation factors, you have to capitalize the letter F in the word factor, and then you have to put next to it the symbol in Latin grammar 1 through 13. You notice that the molecular weight of these compounds varies, and they exist in the liquid part of our blood, in plasma, for various periods of time. The last column on this slide gives you information of the so-called half-life of these particular chemical compounds, which means their existence before they're consumed in some chemical reactions, which are important in the process of the plasma coagulation cascade. This slide summarizes in a flowchart mode the plasma coagulation cascade. You notice that we have two pathways, one of them labeled extrinsic and the other intrinsic. They have different coagulation factors involved, and all of them converge in the center here, which is activation of coagulation factor number 10. The process afterwards moves in only one pathway, and we end at the very end, at the bottom of this cascade, with the formation of insoluble fibrin. The purpose of the plasma coagulation cascade is to form fibrin from a chemical compound which exists inactivated in the liquid part of blood, in blood plasma. The compound is called fibrinogen. As the name indicates, fibrinogen has the ability to form fibrin, and the plasma coagulation cascade has that particular outcome as its purpose in nature, namely to form fibrin from fibrinogen. Let us now review the two pathways of the plasma coagulation cascade. The extrinsic pathway is initiated by the release of what is called tissue thromboplastin. This is a very general term which covers everything, namely all the chemical compounds which are released when the blood vessel wall tissue is damaged as a result of the surgeon's scalpel cutting through it. In the presence of calcium ions, which of course are some of the ions which are dissolved in blood plasma, coagulation factor 7 is activated. Notice how that is indicated. You write down factor 7, and then if you put the subscript A, that means that we have the activated form of that particular coagulation factor. The extrinsic pathway is a one-step process. Immediately after coagulation factor 7 is activated, the process moves to the central part, which is the activation of factor 10. Notice again, factor 10, subscript A, indicating that this particular factor is now activated. Nature has an additional pathway which brings about the plasma coagulation cascade. That is the intrinsic pathway. It starts with activation of coagulation factor 12. What will activate coagulation factor 12 are many things. For example, chemical compounds which are released from damaged tissues, like, for example, collagen, or ADP, released from the activated platelets. And when blood comes in contact with surfaces which are not normal biological, and in this particular slide, that particular type of surfaces is indicated by the word glass, foreign or synthetic materials. Activation of factor 12 activates in turn factor 11. In the presence of platelets and collagen, this particular activated factor 11 activates factor 9. calcium ions are needed in this particular process of the activation of factor 9. With more calcium ions and phospholipids from the activated platelets, and in addition the activated factor 8, we reach the central part of the coagulation cascade, namely activated factor 10. In summary, therefore, starting with either the extrinsic or the intrinsic pathway, two different starting points, two different coagulation factors involved, only one step in the extrinsic pathway, many more steps in the intrinsic pathway, we reach the central part of this particular cascade, which is activation of factor 10. On this slide, let us concentrate on what happens after coagulation factor 10 is activated. In the presence of calcium ions, phospholipid from platelets, and activated factor 5, prothrombin is converted with the help of an enzyme prothrombinase into thrombin. Keep in mind, prothrombinase, as the name indicates, if there is a chemical compound whose name ends in ASE, that is an enzyme. It catalyzes chemical reactions. The important thing about this particular reaction is the following. Prothrombin is a chemical compound which is dissolved in blood plasma, but it's not active. When the plasma coagulation cascade is initiated and reaches this particular level, then prothrombin is activated into thrombin. Nature has put all these steps under control because thrombin is a very potent bioactive compound. In the case of the plasma coagulation cascade, the importance of thrombin is the following. It is the enzyme which converts fibrinogen, another chemical compound which is present, dissolved in blood plasma, but in an inactive form. Thrombin converts fibrinogen into fibrin. This cartoon illustrates very nicely the process of the formation of the fibrin monomers all the way to the insoluble fibrin. You see here the fibrinogen monomers and the peptides A and B, and the way they polymerize now and they stick with each other, if you want to call it like that, is either end-to-end or side-to-side. Up to this point still the fibrin is soluble, but when now this particular side-to-side arrangement is exposed to calcium ion and to the activated factor 13, it becomes soluble. You can see now all these parts are stuck with each other in all directions. Fibrin is formed in a net formation and it has a very special physiological purpose. What is the contribution of the fibrin net to blood clotting? It stabilizes the platelet plug, which now becomes a blood clot or thrombus. A blood clot is shown on this scanning electron micrograph. Pseudocolor has been used and the fibrin net appears as gold or light brown. You can see some platelets caught in the fibrin net, but because the red blood cells are predominant components of blood, a lot of them, although they don't contribute to the process of blood clotting, they are trapped in that fibrin net. I call them innocent bystanders who are caught in this particular process. This artistic rendition gives us another perspective of the blood clot. You see a lot of red blood cells with that characteristic dimple of theirs and the red color flowing by. You can see at the bottom the endothelial cells, and what you see here is the blood clot. Biomedical engineers who have studied blood clot formation under blood flow condition have given us evidence that the clots form this particular tier formation. Namely, you have a body right here, and you can see the fibrin net now stabilizing whatever is trapped inside that net, and then you have like a tail, the tier tail, as blood flows in the direction into the depth of this particular slide. What happens when a blood clot is formed? What is the fate of blood clots inside the human body? Of course, once formed, the blood clot can grow, it propagates, and in addition, it's organized. Remember, we started with a platelet plug which was not very stable in the dynamic environment of blood flow, but now with the help of the fibrin network, it is all stabilized and stays on the location of the vascular tissue damage. What contributes in these processes is what I call synergism. That means how one process helps the other and vice versa. From the perspective of a biomaterials biomedical engineer, we're going to put in the center of these processes of synergism the activating surface. What is an activating surface? It is the damaged endothelium or the inner part of the vasculature which the flowing blood, specifically the flowing platelets, recognize and start the formation of the platelet plug, and the plasma coagulation cascade gets activated and forms the fibrin net. The activating surface could be any synthetic biomaterial that you and I are designing, formulating, and plan to use as part of an implantable device. Once blood comes in contact with this either naturally occurring, or I should say by that damage naturally occurring, or the synthetic activating surface, what coagulation factor will get activated? Of course, factor 12. And once that particular factor is activated, through the speaking of the interaction between the two pathways of blood coagulation, we have the formation of fibrin. But as soon as that particular activating factor 12 comes to be, we have some other very important, intriguing interaction happening. We have the activation of the kallikrein system. The kallikrein system and fibrinolysis on the other side activate the complement system. The complement system, protein activation, activates white blood cells through the process of chemotaxis. So what we have here in the schematic, and then I'll show you again the slide of that, what I call, as I told you before, the pit of snakes, to see how all these processes affect one the other, and they proceed to bring about very important physiological outcomes. As I said, activation of factor 12 in this particular system up here, factor 12 or Hagemann factor, you see it's very close to the green serpentine notion, which is the high molecular weight kininogen prekallikrein system, which brings about the sensation of pain in the body. And that one, of course, is associated with the fibrinolytic system in red. And you can see the thrombin at the very end of the blue system of the plasma coagulation cascade affects and is it related to the process of fibrinolysis? And last but not least, you can see all these little finger projections from the blue serpentine ocean that bring about the yellow system, which is the complement proteins whose activation brings about activation of white blood cells, function of the white blood cells, involvement of the white blood cells in these processes. I want to make sure that we don't miss two other aspects of this synergism. One of them is the effect of thrombin on platelet function. Remember, thrombin is produced through the processes of the plasma coagulation cascade. It's the one step before the very last one, which means conversion of fibrinogen into fibrin. Thrombin, though, when it's released, affects platelets. It activates platelets. It activates them at the stage of the release of the chemical compounds from the granules inside their storage pools. And notice that some of those are fibrinogen and calcium and other ions. It induces aggregation of the platelets, which means the platelets now stick with each other to form the platelet plug, and subsequent release and retraction of the platelet plug. Another important synergism is that of the contribution of platelets, the activated platelets, to the plasma coagulation cascade. You notice that platelets are shown on key points of the intrinsic plasma coagulation cascade, namely in the activation of coagulation factor 12, the coagulation factor which is activated when blood comes in contact with all synthetic materials. Furthermore, platelets are involved in the activation of the next step of the intrinsic pathway, activation of factor 11, but phospholipids, which of course are contributions from the activated platelets and aggregated platelets, are involved in the activation of factor 10 and later on in the conversion of prothrombin into thrombin. I want to emphasize that the processes which we reviewed so far are part of the normal physiological processes which are needed to maintain what we call hemostasis. By that we mean that the body has to conserve the valuable commodity of its blood supply, the 5 liters of it. They have to remain inside the cardiovascular system. The human body cannot afford to lose blood, either because of an injury or trauma, and therefore blood being lost outside the body, or when blood oozes in parts of the body inside which are not supposed to have blood, for example, the abdominal cavity. These are the good aspects of the formation of the blood clot or thrombus. The question now is the following. Once a clot starts, and as we said, it can grow, propagate, and organize itself, does that process go on indefinitely? And if that is so, why aren't we, after we cut our finger or we had some other injury, we haven't become what I call a solid piece of a blood clot? Nature has checks and balances in many aspects of its processes, and you've reviewed some of those in your physiology course. But in the case of blood coagulation, there are some very important checks and balances which we'll review now. In normal physiology, formation of blood clots or the process of thrombosis is controlled through some very important processes which we'll review now. In this particular cartoon, there is an illustration of the process of antithrombin 3 heparin complex. In the liquid part of human blood plasma, there is another chemical compound which is called antithrombin 3. As the name indicates, this particular chemical compound interacts chemically with thrombin and inactivates it once thrombin is formed through the process of the plasma coagulation cascade. Unfortunately, this particular process is very slow. However, nature has put another faster mechanism for inactivating thrombin once the purpose of its existence has been achieved, namely formation of the fibrin network. The process involves another chemical compound which is dissolved in our blood, which is called heparin, and its chemical structure is shown in the insert here. Once antithrombin 3 and heparin form a complex, that particular complex inactivates thrombin in a chemical reaction which is very, very fast, therefore very efficient. Does the antithrombin 3 heparin complex inactivate only thrombin? The answer is absolutely not. The effectiveness of that particular chemical complex is really remarkable, and it's shown schematically on this particular slide. Yes, the complex antithrombin 3 heparin inactivates thrombin down here at almost the end of the plasma coagulation cascade. But notice, in addition, it inactivates activated factor 7. What does that mean? If the extrinsic pathway has started in terms of activation, it's completely blocked now. In addition, the intrinsic pathway, which has very many more steps, it is hit at all key points. Activated factor 12, activated factor 11, activated factor 9. That takes care in inactivating the intrinsic pathway. We reach now the central part of this process. Activated factor 10 is also inactivated by antithrombin 3 heparin. Therefore, the whole plasma coagulation cascade is inactivated when antithrombin 3 heparin is formed. The effect of the inactivation is impressive, and keep in mind, this is a process which prevents blood clots from forming. By stopping or inactivating thrombin, that means the platelets are not going to get activated either. So this is a very effective way of preventing blood clots from forming. The process of inactivation of the plasma coagulation cascade and of platelet activation via the effects of thrombin are preventing blood clot formation. Once the blood clots are formed, that particular process has absolutely no effect. The blood clot, once formed, will stay there and will not be affected by the heparin antithrombin 3 complex. What happens to the blood clots once they are formed inside the vasculature? In the dynamic environment of flowing blood, a part of the blood clot may chip off and flow downstream. Alternatively, the whole clot may dislodge itself, break away from the site of formation on the location of the vascular tissue damage. one result of this particular step is that the subendothelial structures are exposed again to flowing blood, activating platelets, activating the plasma coagulation cascade, and a new blood clot may form again at the site of the original injury, and the particular vicious cycle of formation of clots, dislodging them and moving them downstream will continue. Another major complication which gives rise to pathological clinical conditions is the fact that these particular dislodged blood clots or pieces of them can lodge themselves in smaller diameter blood vessels and form what we call embolus or emboli. Embolus in the singular, emboli in the plural. Lodging of emboli in smaller diameter blood vessels stops blood flow downstream. Therefore, the parts of the body, the tissues, the organs, which depended on that particular blood flow for the nutrients and oxygen, removal of carbon dioxide and metabolic waste, do not get the benefit of that particular function. Therefore, they become deoxygenated, and if that condition lasts much longer, they may die. Even the stabilized blood clots after the formation of the fibrin net may give rise to blood circulation problems. As illustrated in the cartoon of this slide, the protruding stabilized blood clot diminishes the diameter, the internal diameter of the blood vessel, and therefore the blood that needs to go by has to negotiate a much narrower pathway in order to proceed downstream. Such complications give rise to several pathological blood circulation related problems in the hospitals and clinics. Such pathological problems are illustrated on this slide. You can see on the artistic rendition of the heart, the outside part illustrates the blood vessels which bring blood to the muscle of the heart, the coronary arteries, and on one side you see that the artist removed the cover of the blood vessel, and what you see inside there is a blood clot which has lodged itself, and therefore what happens is that now the tissue of the heart beneath that particular spot, downstream from that particular spot, slowly but for sure is going to be denied its oxygen and its nutrients. And as illustrated on the figure on the right hand side of this particular slide, what you have over here inside is a blocked artery, and therefore slowly but for sure the heart muscle which receives the oxygen and the nutrients and removal of the metabolic waste and carbon dioxide beneath that particular spot of the clot will start being in desperate situation and will have eventually a major damage. That particular condition, a situation, leads to what we call heart attacks. And once heart muscle is damaged, it cannot repair itself, and it always looks discolored, and in addition, and most importantly, it is dysfunctional. If it is a very big section or percentage of the heart muscle, the compromise of the circulation and the function of the heart in pumping the valuable blood in the circulation of the human body is really in very big jeopardy, and it may cause serious pathological conditions. In addition to the example of a blood clot in the coronary arteries of the heart, which leads to heart attack, we have similar examples again of clots which are formed in the carotid arteries, the ones that bring the blood to the brain, and when that happens, namely a blood clot lodges itself in the carotid arteries, then we have strokes. If blood clots happen in the veins of the lower extremities, the legs, those conditions result in phlebitis. To make the long story short, blood clots in blood vessels of the body, when they are lodged and they are not removed, can cause major pathological problems and conditions. The last part of this lecture will review what happens when the role of blood clot formation is achieved. This process is called fibrinolysis, lysis of fibrin, and therefore dissolution of blood clots. Fibrinolysis aims at the breakdown of the fibrin net, which has been formed as a result of the plasma coagulation cascade, which we reviewed earlier in this lecture. The process of fibrinolysis requires a chemical compound called plasmin. This particular chemical compound is very bioactive, therefore it doesn't exist in the human blood plasma. It is produced from its progenitor, the so-called plasminogen. As the name indicates, it has the ability to generate plasmin. This plasminogen is present in human blood plasma. It's activated via the action of certain chemical compounds. For example, tissue activator, urokinase, streptokinase, etc. Once plasmin is formed, it breaks down fibrin into smaller pieces which are called fibrin degradation products. However, once the purpose of plasmin has been achieved, namely to break down the fibrin network, it is inactivated via the action of a chemical compound which is present in the liquid part of blood plasma, and this chemical compound is called antiplasmin. This slide summarizes the conversion of plasminogen into plasmin, showing that various chemical compounds like streptokinase, urokinase will convert plasminogen into plasmin, and once plasmin is formed, it will break down fibrinogen and fibrin into what they call fibrin degradation products, as we saw in the previous slide, or fibrin split products. The important detail of this process is illustrated over here. We start with fibrinogen and fibrin, chemical compounds with molecular weights, let's say, around 330,000. The action of plasmin breaks them to what they call fragment X. This particular fragment is still very big, molecular weight 240,000. More action by plasmin converts fragment X into fragments Y and D, respectively 150,000 and 80,000 molecular weight. More action on fragment Y, because it's still quite big, gives us fragments E and D with molecular weights 53,000 and 80,000 respectively. When the fragments reach the point of 80 and 53,000 molecular weight, they are small enough and they can be removed from the body through appropriate and pertinent normal physiological processes. The purpose, therefore, of the lysis of breakdown of the fibrin network is that in that way it is achieved. The video Hemostasis Helpful Blood Clotting is an overview of what we've discussed so far. It was shown here, and in addition I'm giving you the link to this particular video so you can watch it at other times at your leisure.
Normally, blood clots form in response to injuries which cause bleeding. Clot formation helps to conserve the body's supply of blood so that small cuts don't end up draining the body of all of its blood. The body's process for stopping the bleeding by forming a clot is called hemostasis. In this illustration, the blood vessel contains red blood cells, white blood cells, platelets, and tiny proteins called clotting factors. These components of the blood float together in a straw-colored liquid called plasma. When the vessel wall is torn open by an injury, blood begins to escape. In response to this damage, the vessel quickly constricts, reducing the flow of blood. This constriction lasts only for a few minutes as the body puts in place a plug to stop the bleeding. Platelets soon recognize the injury and begin to stick to the damaged surfaces. This is called platelet adhesion. The adhering platelets change shape and release chemicals which keep the vessel constricted and draw more platelets into the damaged area. These platelets are said to be activated. Additional platelets arrive and begin to stick to one another so that a loose plug is formed. This is called platelet aggregation. When tissue is torn apart during an injury, blood is exposed to a chemical called tissue factor, which naturally resides on the surface of tissue cells, or may also be displayed on the inner lining of the blood vessel wall. This tissue factor sets into motion the chemical reactions of the clotting factors found floating in the nearby blood. Around a dozen different chemicals called clotting factors circulate inactively within the blood. When called upon, these clotting factors participate in a complex sequence of chemical reactions to ultimately produce tiny strands of a strong material called fibrin. Fibrin strands form in and around the loosely arranged plug of platelets, and eventually form a meshwork which tightly binds the plug together. This meshwork keeps the platelets from being washed away by the flow of blood and makes the platelet plug more watertight. Red blood cells and white blood cells become trapped in the meshwork as it forms, giving the plug its characteristic red color. The overall process of forming these fibrin strands to solidify the clot is called coagulation. So within a matter of minutes, a sturdy plug is formed to arrest the outflow of blood. But too much coagulation activity allows the clot to grow much larger than it should. Two groups of the body's own naturally produced chemicals help to limit the size of the clot. The first group blocks coagulation. These substances are known as coagulation inhibitors or natural anticoagulants. When these chemicals interfere with the reactions among the clotting factors, fibrin formation is blocked, and no new fibrin strands are available to hold additional cells to the clot. In this way, clot growth is kept to a minimum. The other group of chemicals keeps clot growth under control by cutting the fibrin strands and actually dissolving part of the clot. These chemicals are part of the natural process known as fibrinolysis. Fibrinolysis occurs as chemicals called plasminogen activators are slowly released from the inner lining of the damaged vessel wall. These plasminogen activators trigger the destruction of the fibrin strands that hold the clot together. So coagulation inhibitors can only stop the growth of the clot, but fibrinolysis can actually dissolve part or all of the clot. In fact, when the vessel is completely healed, the remaining clot is dissolved by means of this second group of chemicals. To sum up then, rapid clot formation is essential for conservation of the body's supply of blood. We've seen the constriction of an injured blood vessel, the formation of a platelet plug, the strengthening of that plug by the addition of fibrin strands, and the body's protective systems, preventing excessive clot growth.
The video Thrombosis, Harmful Blood Clotting is also an overview of part of the aspects which we have been discussing in this course so far. The video is shown here. In addition, the link to this video is provided on this slide for your information.
Blood clots are helpful when they form to stop the flow of blood from an injury, but blood clots can also form in harmful ways at unexpected times and in vessels that have not been injured by outside trauma. Even helpful clots that form to stop the flow of blood from an injury may become harmful if they expand in the vessel beyond their beneficial dimensions. In 2 prior videos, we examined helpful blood clots, how these clots form in response to injury, and how these clots are replaced by scar tissue in the natural healing process. In this video, we'll take a look at harmful blood clots and what naturally happens to them after they've formed. The formation of a blood clot, where the clot may be harmful instead of protective, is called thrombosis, and the clot itself is referred to as a thrombus. When these clots occur in arteries, they may result in conditions like stroke and heart attack, and when they occur in veins, they may produce deep vein thrombosis, pulmonary embolism, and rare conditions like retinal vein occlusions. In a separate video, we'll consider the circumstances that might cause a harmful clot to form, but right now let's look at what might happen to the clot after it forms. In this illustration, the blood vessel contains red blood cells, white blood cells, platelets, and clotting factors that float together in the liquid part of blood, the blood plasma. If we add a little more detail to these vessel walls, we see a layer of endothelial cells forming a 1-cell thick interior lining for the vessel wall. This lining is called the endothelium. One of the important functions of the endothelium is to prevent blood cells from sticking to the vessel walls. In this example of thrombus formation, the absence of the protective endothelial lining sets the stage for platelet adhesion and activation. Additional platelets and other blood cells are bound together by a meshwork of fibrin strands, and a substantial thrombus is formed. So after a potentially harmful clot begins to form, what naturally happens to it? What are the possibilities? There are 4 main possibilities: dissolution, propagation, embolization, and organization, and we'll go through each one of these. One possibility is that the clot may completely dissolve on its own. The body's natural fibrin severing process, fibrinolysis, may be enough to completely dissolve the clot. But even if the clot is only partially dissolved, blood flow through the vessel may be significantly improved. Another possibility is that the clot may continue to grow or enlarge. This expansion is referred to as propagation. Propagation takes place when the body's natural coagulation inhibitors are overwhelmed by the stimulus for clot formation, and the body's natural fibrinolysis response turns out to be inadequate. Over time, a clot may grow completely across the opening of the vessel to block off the flow of blood through the vessel. We'll show a cross-section here over to the right to give a better idea of what's happening. This is called an occlusive thrombus and is more likely to occur in medium and small-sized vessels. In addition to expanding across the opening of the vessel, clots may also grow lengthwise inside the vessel. In many circumstances, a clot will grow in the direction of the blood flow. In other circumstances, the clot may expand opposite to the direction of blood flow. When a thrombus propagates in an area of fast-moving blood flow, it may contain a greater proportion of lighter-colored fibrin and platelets, and a smaller proportion of red blood cells. This gives the thrombus a pale, firm appearance. When a thrombus propagates in circumstances where blood flow is slower, the proportion of red blood cells trapped within the expanding thrombus is much greater, giving the thrombus a deep red color. A third possibility is that a part of or the whole thrombus may break off suddenly and be carried away by the flow of blood. This is said to be an embolization of the thrombus. The portion of the thrombus that is carried away by the flow of blood is called a thromboembolus. If this happens in the left side of the heart or inside of an artery, the embolus travels away from the heart along with the flow of oxygen-rich blood and gets stuck when the size of the embolus exceeds the interior dimension of a progressively narrowing artery. An arterial embolus may become lodged in any number of locations, including the arteries supplying blood to the heart, the brain, or the kidneys. If the embolus blocks the supply of oxygen-rich blood to a tissue for a sufficient length of time, the tissue located downstream of the blockage may die. If part of a thrombus breaks free inside of a vein, the embolus travels back toward the heart along with the flow of oxygen-depleted blood. The embolus travels through the heart along with the oxygen-poor blood and exits the heart on its way to the lungs, where the blood will absorb fresh oxygen. But the vessels that branch out from the heart into the various areas of the lungs are progressively narrowing, and at some point along the way, a venous embolus will get stuck in one of these vessels. This results in a condition called pulmonary embolism, or more precisely, a pulmonary thromboembolism. If the thrombus remains in place without being dissolved, the body begins to treat the thrombus as if it were a wound that needed healing. The wound healing process was introduced in a prior video of this series entitled Wound Healing. Similar to what was seen in wound healing, white blood cells within the thrombus, including macrophages, begin to engulf and digest any cells of the thrombus that may die off. Fibroblasts and capillaries, together in a tissue called granulation tissue, slowly invade the thrombus. This migration of cells and capillaries originates from the vessel wall and travels through the point or points where the thrombus is attached to the vessel wall. Parts of the thrombus where the invasion of granulation tissue is well established are said to be organized. Once an area of thrombus is organized, that area will not be susceptible to dissolution by fibrinolysis. But complete organization may take a matter of weeks, and while one part of the thrombus may be organizing, other parts may be dissolving. The cells of the granulation tissue begin the process of degrading the thrombus and replacing it with fibrous scar tissue. If a thrombus only grows large enough to partially block the opening of a vessel, when the clot becomes organized, the resulting scar tissue may eventually shrink and be pulled into or incorporated into the side wall of the vessel. This allows resumption of greater blood flow through the vessel. If a clot grows to completely block the opening of a vessel, as the clot is organizing, elements of the granulation tissue secrete chemicals which degrade the existing clot, leaving open spaces or pockets within the thrombus. These pockets become lined with endothelial cells. They are thought to gradually link up such that small channels are created from one end of the thrombus to the other. The formation of these tiny passageways through the clot is called recanalization. Recanalization may begin as early as a few days after the occlusion of the vessel, and over time the initially small channels may expand to accommodate more blood flow. To sum up then, blood clotting is normally a helpful process that can work to preserve the body's supply of blood in the face of injury. But blood clotting apart from injury or as an overresponse to an injury can be very harmful. In this video, we've looked at some of the aspects of harmful blood clotting, with a special emphasis on what can naturally happen to a thrombus after it forms. We've seen that the thrombus may totally dissolve, or continue to grow, or break off and be carried away by the blood flow, or replaced with scar tissue and eventually be pulled into the vessel wall, or be pierced by tiny channels.
This is the end of Lecture 9. the blood plasma coagulation cascade, blood clot, also known as thrombus formation, and fibrinolysis.